cloning with PBR322. How we can clone foreign DNA by using this vector molecule. As I told you that plasmid, if we are going to use them as a cloning vector, it must have certain desirable properties. तो अगर हम इसको देखें PBR322 को, तो इसमें ज़्यादातर जो प्रॉपर्टीज़ हैं वो पाई जाती हैं, लाइक कि इसका जो मोलेकुलर वेट है वो कम है. If its molecular weight is low, that is around 4361 base pairs, so mostly it is present in high copy number. So copy number may be 20 to 30 per cell. And if we treat the recipient cells that contain PBR322 with chloramphenicol, as we discussed that the copy number, due to inhibition of protein synthesis, it can be further enhanced, like it may be 1000 to 3000 copies per cell, so that the cloned gene, it can be overexpressed, because we can have number of cloned fragments of our desired gene that may represent up to 40 to 50% of the nucleic acid of the cell after treatment with chloramphenicol. Another desirable property is that during isolation, mostly this plasmid, it is present in supercoiled form. So its isolation, it is relatively easy. And moreover, the DNA fragments that is having the length of 10 kilobase, they can be cloned by using PBR322. Another advantage is that the whole sequence of PBR322, it is known. So we have the idea that how many type of restriction sites are present and what are their locations. And moreover, some selectable markers in the form of ampicillin resistance and tetracycline resistance, they are also available. And additionally, single site for different type of restriction endonucleases. So all these traits, they are highly desirable that are present in PBR322, so it can be used as a cloning vector. And later on, after transformation, the recombinants can be screened on the basis of ampicillin or tetracycline resistance. How? So this diagram, it indicates the use of PBR322 in cloning. So here we have number of plasmids, that is PBR322, that contain ampicillin resistance gene or tetracycline resistance gene. Suppose we have cut or digested this plasmid or even the foreign DNA with restriction enzyme, that is Ph.D1, and this Ph.D1 site, it is present within ampicillin resistance gene. So both the foreign DNA and the plasmid DNA, they have been digested, and later on they will be ligated. When DNA will ligate within the region of ampicillin resistance gene, so its function will become inactivated. So that cell will become sensitive to ampicillin but still resistant to tetracycline. In the next stage, the recombinant molecules or plasmids that are produced, they are introduced by the process of transformation into the E. coli cells, so that our host organism will contain the plasmids that may contain foreign DNA or may be recircularized, that we will screen in the subsequent steps. So plasmids either with foreign DNA or without the target DNA, they are inserted. After transformation into E. coli cells, all the cells, they will be grown on a medium that is supplemented with tetracycline as a selectable marker. because ampicillin gene, it is inactivated by the insertion of the foreign DNA, so that cell will become sensitive to ampicillin. So that's why in first step, all the transformants, as they are resistant to tetracycline, they will be grown on tetracycline supplemented medium. And at this stage, there are chances that the plasmid, it may be recircularized or it may have insert of foreign DNA. Because in either case, the transformants are resistant to tetracycline because its function, it is not inactivated. Then how we will know that our gene of interest is present? In the subsequent step, the transformants, they are plated on two media, one that is supplemented with tetracycline that will act as a control, and other that contain ampicillin as well as tetracycline. So all the cells, they will grow on tetracycline medium, and on the other hand, the medium that is supplemented with ampicillin plus tetracycline, so only those transformants that contain the foreign DNA, they will be sensitive to ampicillin. But tetracycline pressure, it is also there, so they will only grow here. So only those microbes that contain foreign DNA, they will be able to survive on tetracycline medium in the presence of ampicillin. So this is the strategy by which we can select our transformants by using PBR322 as a cloning vector.